Yeah. Yeah, you can start introduction.
Yes, sir. A very good evening to Kannan sir, Shrushti, and all my dear students. So we have gathered here this evening to have a wonderful talk by Kannan sir. So let me give you a short introduction of Kannan sir. Professor Kannan Krishnamurthy was one of the first faculty member to join as a reader, associate professor in biotechnology at the University of Pune, that is Savitribai Phule University, under the MSc program, MSc Biotechnology program, MBTB, initiated by Dr. S. Ramachandran to create manpower for the emerging area. He was the former Vice Chancellor of Nagaland Central University, the first professor and founder, Dean of University School of Biotechnology, Guru Gobind Singh Indraprastha University, and the first convener of biotechnology for CBSE school in 2002. He is well known as a protein chemist who established facilities at CFTRI and CCMB. Was also a director at SPAN Research Center, Surat, where he explained the mechanism of bone marrow transplantation and GM, and associate director of biotechnology at Ranbaxy Research Lab. He developed the process for making whey protein concentrate for Mahan Proteins and fish technique for DSS image technique. He is currently the founder of KK Biotech, which is a registered company promoting biotechnology. It is in New Delhi. I welcome you, sir, formally. The session is, I declare the session open. And, sir, over to you.
Yeah.
Yeah.
I just thought first, I'm going to go through the questions which was sent to me. Then we will begin the whole session. Okay. So,
Sir.
just sorry for this small. Yeah. So thank you for agreeing to be part of this session. And I'm so glad so many of you have registered from the first year. And let me begin with the kind of questions which are raised. Okay. Um, First question, which I was surprised coming from my first year, that which companies do we have chances of getting a job? How much salary? What career path? Which field in biotechnology? Whether it will benefit PhD was the question. What is more beneficial, a BSc or a MB or MSc? What are the challenges you are facing? What publications? How do you balance creativity? How do you, in which field of biotechnology can we proceed further to have our own startup business? What type of skills does a person need? Apart from India, which other country there is scope for after BSc biotechnology? Except research, is there any, can we work in a sales department, etc., etc. So there are a lot of questions which have been asked, and there are about 25 of them which I saw in this slot. Okay. So to answer these questions, I must give you a basic idea of what we are seeing today. So are you ever puzzled by this questions which I have raised, which you should respond? What is your opinion on bioscience, biotechniques, and biotechnology? Does it make any sense to any one of you? Are you doing bioscience? Are you doing biotechniques? Or you are doing biotechnology? Anyone would like to look into that point? Is it a valid question to ask? Children,
Yes, sir.
have you ever wondered?
Yes, sir.
Okay, that's good. Now, what is your definition of biotechnology? So I used to often wonder, and I coined something for the school children way back in 2002. I said, and I'm going to give you the definition which you can carefully listen. I said a biotechnologist is a mathematician who does addition, subtraction, multiplication, and division of biomolecules in a cell. Just to elaborate, what do I mean by addition, subtraction? Today, diabetes is a major problem all over the world, including India. So we need insulin. Earlier, we used to get this insulin from pigs. From the pig pancreas, we used to isolate. Okay. For one man's requirement for a year, we nearly need to kill about 106 pigs. Okay. So today, our estimate is something like, large population, something like 50 million people are suffering from diabetes in India alone. So if you were to make it from pig, so you can multiply, 50 million with 106, and you can see the population of pigs will be more than the population of the whole world. So this was a challenge. Okay. So what did they do? So there were two technologies which came in. That's why I'm saying there are two types of biotechnology which you should be aware. One is called the classical and the other is called the modern biotechnology. Okay. Now, classical means you take out products which the cell is already making. Like, for example, say penicillin. So the penicillin producing microorganism, or Penicillium notatum, it was already making, only we learned to take it out. We knew it is secreted, so we learned how to do it. Then we look for a better strain which gives, and then penicillin was made. But if you look at the microorganisms, they did not make insulin. So what we did in the modern biotechnology is we learned what is called genetic engineering. That is, we take out the gene from a source for insulin, that is subtraction. We add it to a vector, which is called addition. We make photocopies of it, which is multiplication. Then sort out the cells which are making insulin from which are not making, that is called division. So this is all what we are doing as far as the genetic engineering is concerned. That is, you have made an organism do something which it was never doing before. And because of that, we could, from 1978 onward, insulin became available in the recombinant DNA form. I also want you to know that even though recombinant DNA was discovered way back in 1973, there is a lot of worry whether we'll disturb the environment, okay, and we'll produce some organism which is this. Like this is what they are talking about Covet, that probably it is a man-made organism. So this was a worry even those days. So there was a total ban on making, using recombinant DNA, till scientists met together and found out a guidelines how to make a safe product. After that only everybody started using laminar flow hoods. So you would have seen what is. So laminar flow hood makes sure things don't come. And if it is a very dangerous one, now we have what is called BSL1, BSL2, BSL3, like that. There are special facilities depending upon how dangerous the organism is. So modern biotechnology is one where we are doing this. So we are making a product by a totally different way. Okay. So when we look at this, there is another. So, and this was a patented technology. Okay. So if anybody couldn't make insulin, the royalty, the whole thing was with the, the patenters, and they gave it to a company called Genentech, who made this product in 78. Since they couldn't market it, they gave it to a very big company which marketed it. and both University of California, San Francisco, and Stanford University scientists got the royalty for it. And the university alone got about nearly $1 billion as royalty for selling. So this is the first time the university started benefiting, because at that time when biotechnology started, nobody wanted to invest money. So Genentech said, look, you can use your laboratory facility in your university, make the product, we'll pay you for using the facility. So that's how the whole thing happened. Another company which became very famous is Amgen, which produced a product called erythropoietin. So if anybody asks you, which are the hottest selling products to begin with, they are erythropoietin and insulin, which became the big one. Okay, so there is a big excitement in this. But there is another technology which developed, which is called Hybridoma Technology. Okay, which was developed by two scientists, Cesar Milstein and George Kohler. Now, if you look at that, they did not take a patent. They refused to take a patent. So when they developed it in 75, it was freely available. And within a year, there were 200 companies in USA alone, which are making antibodies, what is called monoclonal antibodies. Okay. Now, so suddenly, because of not patenting, it became, and you will see that the whole diagnostic industry boomed because of that. Okay. So modern biotechnology had two major ones. One is genetic engineering, another is hybridoma technology. Okay. And because I want you to know all this, because biotechnology is not that old. The modern biotechnology is not that old. So when you say job opportunity, etc., that industry has to develop. So a countries like India, because we cannot violate the patent, it took us many years to get into that. So we only made products which are off patent, because the patent goes off in 20 years now. So when the patent is about to be over, people make it. That's how. Even in pharma business, all are what we call as generic products. Okay. Then what is the difference between biochemistry and chemistry? See, the biochemist, the biggest thing is they use what is called a buffer. Chemists don't use buffer. They use organic solvents and things like that. Now, just to give you one very interesting example, when penicillin was initially made, it was only soluble in organic solvents. But the moment you extract an organic solvent, its shelf life was very low. Its activity disappeared in a few days. So a man called E.B. Chain, who got the Nobel Prize along with Alexander Fleming, who was a biochemist, he extracted an organic solvent and dumped into a buffer at pH 6.5. Suddenly, penicillin became very stable. It was as stable for 3 years. So because a compound is stable for 3 years, you could make plenty of it. And USA was the forefront, everybody there. Just like in COVID, USA spent money like anything to make the vaccine. They tried all vaccines. There are about 198 vaccines still going on. 150 are already in clinical trial. So USA, when it gets into some big this thing, they go in all out, and the government supports the industry. So what we see here is that the penicillin story was the first one where it was stable. So the advantage was soldiers were dying not because of the bullet, because of infection. They were all given penicillin, so they all survived. That's why it's called the miracle drug of the 1940s. Okay. Then then antibiotic era happened. So everybody was looking for new drugs, and one of the most famous is Aureomycin, who they discovered was an Indian by Elapragdas Barrow, his name. So he developed the broad spectrum antibiotic, tetracycline and all are out of that. Okay. Another big thing is, how do you measure? For example, say glucose and all occurs in large amounts, so we are able to measure it by colorimetric methods. Okay. Now you must know that to see color, we need something like 10 to the power 16 molecules per ml. But hormones occur at 10 to the power 12. So we had to develop a total this thing. Some other time I'll talk how we managed to measure the insulin. And this was done by Rosalind Yallow and Burson, and who developed what is called radioimmunoassay. Okay. Then another very important thing is, what is the difference between treating a disease, curing a disease, and a disease becoming free? Today, there are some methods whereby, for example, somebody has a genetic disease called thalassemia or sickle cell anemia. By a technique called bone marrow transplantation and using hematopoietic stem cells, we are able to make the person disease free. They don't need any more treatment. Okay. Now, the biggest one is, what did we learn from the COVID-19 pandemic? Okay. Now, that is the interesting one. Okay. Now, just to give you an idea, we have a department called Department of Biotechnology, which was created in 1986. It originally started as a National Biotechnology Board way back in 1982. And the member secretary was a man called Dr. S. Ramachandran, who became the secretary in 86. So if you can see, because we are dealing only with a cell, we don't care from where the cell came, whether it came a plant cell, microbial cell, animal cell, human cell, we don't care anything. So you will see that how to handle these cells and do the genetic engineering or whatever you want to do. So you will see that because it is what we call as a platform technology. It can be applied to agricultural plant sciences. We can apply to plant biotechnology and bioresources, medical intervention, infectious disease biology, human genomics, cancer, computational biology, chronic diseases and human nutrition, and then energy and environment, animal, livestock, allied sciences. So you will see that when people ask me, where do I have an opportunity? You have opportunities in all of them. So this is the government's focus. So I suggest that you visit the website of Government of India called dbtindia.gov.in. And you must patiently go through them to know what is happening. Okay. Now, as you can see, government has also created institutions. One of the earliest one was National Institute of Immunology. Then we have what is called, it originally started as Animal Tissue Culture Facility. Now it is called National Center for Cell Sciences in Pune. We have National Brain Research Center in Gurgaon called Manesa. Center for DNA Fingerprinting and Diagnostics in Hyderabad. You have National Institute of Plant Genome Research in Delhi, Institute of Life Sciences in Bhubaneswar, Institute of Bioresources and Sustainable Development Northeast, in Gurgaon, in Faridabad, in NCR of Delhi. You have Translational Health Science and Technology Institute. Then you have Stem Cell Science and Regenerative Medicine in Bangalore. In Faridabad, you also have Rajiv Gandhi Center for Biotechnology. So when you say you interested in immunology, go to NII. If you want to do cell sciences, go to NCCS. If you want to do brain research, this thing. So you should be aware that these institutions do exist. Okay. So this is the first thing you must know. Okay. And then there are also public sector units which was started those days. What is called BIPCOL, Bharat Immunological and Biological Corporation. That is very near Delhi. Then you have Indian Vaccine Corporation Limited. And the third one is Biotechnology Industry Research Assistance Council. Many of you have done, passing out to a BSc Biotechnology, you can get a scholarship. You apply to BIRAC, okay? And or there are so many others also you have, where you can, go for an internship where the government will pay you money, okay? So there is also, if you have a project proposal to do, you can go to BIRAC for funding. The private party will put 50% and the government will give the 50% money, okay? DBT also has international collaborations in Europe, Belgium, Netherlands, Denmark, Germany, Switzerland, Spain. In Africa, we have a tie-up with South Africa, with Australia. We have in Europe, we have UK, European Union, France, Sweden, Finland, Norway. North America, South America combined is called America. So you have tie-up with Brazil, Colombia, Canada, USA. And in Asia, we have tie-up with Japan and Russia. So you should be aware of what are the avenues available. Okay. Then you also have multilateral NGO corporations and outreach programs. Okay. So you can have multilateral engagements with G20 countries, BRICS countries. Foldscope is something, please go and search in the net. They provide very simple microscopes and other techniques for a low cost so that you can do this thing. CRUC is cancer research under the UK program. So if somebody is interested in learning about cancer research, you can join into that. All this is given in the website of the Department of Biotechnology. Okay. And similarly, you have outreach programs to popularize biotechnology. So I strongly recommend all of you should go and see the biotech site. Okay. And then how to manage projects. So there's a project management system. The government does everything, only you have to look around. They also have what is called Societal and Star College scheme. So it is better because they are it is a UNESCO program and they are trying to improve the STEM, that is science, technology, engineering, and mathematics. So if you look at all these things, you will suddenly see that you have to know what do you want, and there is somebody doing it, and the government is there to support you in a very, very big way. So when you are doing BSc first year, I strongly recommend we can form a club. If all of you, we can have a membership kind of a thing, so that we can meet every once a month or 2 days in a month, and then discuss and say, okay, then I can put you in touch with somebody from whom you can learn. Okay. So this question is, what do you want? You must decide when you are in the first 3 years. Start planning what you like, what you want to do. And when you are coming to the final year, by that time, because if you are part of, interacting with me, by that time, we'll be able to crystallize what you want. Just to give you an idea, the Minister of External Affairs, Jai Shankar, he is my contemporary in college. There was a whole group which wanted to become a bureaucrat, IAS or IFS. So from very first year, they were preparing as a team. So in the final year, they go and appear in the IAS and clear in the very first attempt. So you have to start planning from very first year. And fortunately, you have time. If you take, even if you spend half an hour a day as an extra time, I can help you to build up your thoughts, what all backgrounds you should have. For example, I wanted to be a protein chemist. So from my first year BSc, I learned what is amino acid, how to separate them, how to purify them, how to characterize them, what is a peptide, what is a protein. Everything I learned in my first year, second year, third year. So when I went for my PhD, I was at home. I knew what to expect. So what I suggest, since you being in first year and going into the second year, if it is okay, we can form a small groups and we can decide, okay, these are things you should know, and then we'll decide which area you want to get in, because you have to make a commitment, I want to work in this area. Then the rest can be worked out. Okay. Now, you also say in the process of BSc, you can also learn some skill training. Okay. Another is, should I do an MSc? These are all calls you can take. No, I want to go and work. Because once you have a skill, guy will hire you. MSc or a PhD doesn't matter to the company. They want somebody who will do that job. Okay. Now, questions will come. When you come to that level, we discuss about PhD, what are the issues, what are the opportunity, what are the challenges. Okay. And when I was a kid, I remember how to measure volume of blood in the human body. Because the only thing I knew was measuring cylinder. And if I take out the blood, the patient will die. So it puzzled me. Or my teacher said there are 10 billion RBC in the body. I said, how do you count them? Because if I were to count one cell at a time per second, it will take me more than 31 years. So these are all things. So I'm sure all of you have so many questions in your mind, which you start, when you start answering, you will discover, okay, how do they do it? What technique should I learn, etc., etc. You will learn. In fact, today with the technology, we are able to measure in a single cell how much is the protein. This is what we call a zeptomole. I don't know whether you have ever heard of this word zeptomole, because you have heard of mole, which is 10 to the power 23. Then you heard of millimole, which is 10 to the power 20. Then you heard of micromole, it is 10 to the power 17. Then there is nanomole, 10 to the power 14. Like that, if you go then picomole, femtomole, things like that, they go on. When it becomes 10 to the power 20 minus 23, that is what we mean zeptomole. So what I'm saying, today we are able to measure 600 molecules in a cell. And now we are able to see if there are two cells, one is cancerous, the other is not cancerous, we can study what are the proteins expressed. We can find out which is the protein changing. We can sequence the protein within that cell, etc. So technology has taken so far that you need to become familiar. And this is why you must spend half an hour every day. If you are, part of our club, I can always take you through the journey, how they do it. So today technology has become so high level, and plus with artificial intelligence coming, the machine does everything. So what is your role? So you have to find a role for yourself. So that's what you will see here. Now I want you to understand that, India has been talking of first science policy way back in 1958, which you should be aware of, which laid the foundation for scientific enterprise. Some of you have asked me, can I become an entrepreneur? So you must be aware of the science policy, okay? And today, because of the COVID-19, the fifth policy has come. The government has come out of the fifth national science technology, that is called, STPI, Science Technology Policy of India. 2020, which the final this thing should be coming anytime, to march ahead on a sustainable development pathway to include economic development, social inclusion, and environmental sustainability for achieving Atmanirbhar Bharat. Nowadays we are. So this is the government's decision. Do you want to be party to it, to make India Atmanirbhar? Is something, it is in your hands. Nobody is come and going to tell you do this. You have to discover where your opportunity is, this thing. So there is a greater emphasis. Government is supposing, now you saw for Ayurveda product, Prime Minister has tied up with the WHO, and now they have put $250 million to do clinical research to develop our Ayurvedic knowledge. That's called traditional knowledge system. Okay, developing indigenous technologies, encouraging grassroots innovation. Okay, emergence of disruptive and impactful technologies poses new challenges and simultaneously greater opportunities. Like COVID-19, now all of you know what is the RT-PCR. Before that, nobody knew what was RT-PCR. We used to teach students, but suddenly now even a layman talks about RT-PCR. Okay. So COVID-19 pandemic provided a compelling opportunity for R&D institutions, academia and industry to work in unison for sharing of purpose, synergy, collaboration, cooperation. So when I started this KK Biotech about a year back, so I said, no, I must try to promote, and that's why today I'm talking to you, to promote biotechnology among aspiring and practicing biotechnology, life science, chemistry students, etc. I was also very much, I've had close interaction with His Excellency, the late Abdul Kalam. We used to closely interact. So I always felt he had great visions. He made a Vision 2020 document also. So what I'm saying is, that all of us have to contribute towards the nation development. So what do? We do mentoring of students, so we can do it at an individual level, as a group. We can give you a personalized training or internship, and we can also reach out to you. What a student outreach programs we do have. We help all the level, right from high school children up to bachelor's, master's, Ph.D, postdoc, entrepreneurs, whoever wants. Okay. And so we want to facilitate like your college and all, I would love to facilitate them to grow, okay, in the field of biotechnology. Okay. So if you register with ours, we have a club called Biotechnology Education for students and teachers, called the Best Club. So you can, there we'll be meeting every month, discuss. I'll get you some experts, etc., so that you can interact with them, and then we can have an annual symposium or something where you meet 4 or 5 different from USA, Europe, wherever you want. It can be done. So, then we give certification of practice. Then we can take you to the industry or ask them to keep you for a month. And if you want to understand the subject, we can definitely help you. So that is why I was talking about the Best Club. And I would appreciate, for your college, I can create a separate club so that all of you can interact. Okay. And I strongly suggest you must, this is the vision of this thing: attaining new heights in biotechnology research, shaping biotechnology into a premier precision tool of the future for creation of wealth and ensuring justice, social justice, especially for the welfare of the poor. This is the vision, and this is the vision which the government of India has, which you should be aware. Very interesting that biotechnology, the nodal agency is the DBT, which approves for industries. So they have the permission to grant launching of products, making of products, etc. That is how Bharat Biotech managed to make vaccine, Covaxin. It was totally supported by the Department of Biotechnology. Okay. So I want you to be aware of this. Okay. So we want to realize your full potential, and that is the government's vision also. Okay. And we want to get money put into certain process products, processes, technologies. Okay. We want to improve the efficiency, etc., so that our country becomes self-sufficient. Okay. So there are a lot of things and the government is, and fortunately, somebody I knew is now heading the whole department, who himself did MSc Biotechnology way back in 1987 to 89, who is now heading the whole Department of Biotechnology. Okay. So the core value is integrity, transparency, accountability, teamwork, commitment, excellence, etc. Okay. So I'll stop here, and then you can see, and I leave now the floor open to the students. Okay. Yeah.
Yes, students, if you have any doubts, you can, or if you have any problem, you can ask.
Because this is the time to open up, because you ask all questions. All your questions can be answered. If you visit the website, I can put you in touch with these people who are looking after it, because they're all people I know personally, and I can make you interested. A lot of things can be, but don't ever say, what is my future? The government has invested so much for you, and it is up to you to find out. All of you see many things on the website. I strongly feel you must go through the website, then we can discuss. And if you become a team, I'll help you to start a project, make a product. All these things I've been doing all along. But with students, I would love to do it. But then, as you know, getting people means I'll have to pay them for their talks and all that. So what I'm charging is very minimum. I can tell you that. If you are a group of people, I can even charge you less. But it's not the money for, it is not for profit I am doing it. I said I want to help, but people whom I invite, I have to pay them, because they will come and spend their time. Everybody is not philanthropic that I will do it free. So the money is not, and what we are charging, I can tell you, if somebody wants a personal meeting, I only charge 500 rupees per hour plus GST. So that, it doesn't pinch your pocket so much. So anytime you can talk, we can work out something, put you in touch with people, so that your idea, so that you go with a master plan. And let me tell you, children, I'm very happy you are in the first year, because you have another 2 years. Can we do something for ourselves? Instead of asking, oh, what should I do? Should I do this? Should I do that? That decision you will make yourself. Okay? So you don't have to go through the normal rut of going through BSc, then MSc, then Ph.D. That's one way of thinking. In fact, I just want to tell you, when I started Biotech in Delhi, I started the BTech program, one of the early people to do it. The first question I posed during the counseling is, your aim should be to become a millionaire by 28. So they were stunned. I said, look, then you won't look for a school teacher job, college professor's job, all these things, because you'll never make million. Then you will suddenly realize, I should take a patent, I must make a product and make money. You look at Bill Gates, all of them did that. So why not you? But yeah, it depends on your mental thinking that I can do it. And we are here to realize your potential, because we have taken a lot of risks, so we can help you to, what do I say, help you to nurture your thoughts. So this talk today I'm talking so that I can be of some help to you, because I have gained so much in my life, I would like to share. In fact, you won't believe I have interacted with more than 50 Nobel laureates. So I have a lot of things to share with you. And I can tell you many things, but let us, first I hope now many of your questions are answered. Nishita?
Yes, sir.
Huh?
Yes, sir.
Because there is opportunity. Government has opened the whole thing for biotech. It's a huge investment. And you are not tapping that investment. You are looking for small level. That's why I said, I want to become a millionaire. How can I tap my government to make the best use? How will I get the money out of them? Let them invest the money. I will make the product. So there's a lot of opportunity. Anybody else want to ask?
So, can you
Sir,
please
there are.
talk about the club and membership?
Well, see, I can work out a special package. Normally, individual level, we charge, for a whole year, if somebody joins, we charge 5,000 plus 900 rupees, the GST I have to pay to the government, because I run it as a this thing. So any transaction I do, I have to pay 80% to the government. Okay. So what I am only getting is minus 18%. And that is what I reinvest into. For example, I did an internship for students for a month, okay, online. And they learned how to do mass spectrometry, how to interpret the results, how to do fluorescent activated cell sorter, how do you do cell, what is stem cell biology, what is its application, how to identify a new coronavirus from the sequence. So somebody did a live demonstration, 1 hour theory, 2 hour practical. We did it for a month. Like that, we did a lot of nice programs. And we charged them very less. I mean, for a month's program, because all of them, I paid them peanuts, because that's all what the students could afford. But normally we charge for such programs somewhere around 8,000 to 10,000 for a 1 month full package. But it will be Monday to Friday, 1 hour theory, 2 hour live demonstration. Okay. And you can go to my website, you can see what all we have done. Then we give a certificate, everything. And you can make your report also, what all you learned. So it will be something like a 100-page report. Tomorrow you go for an interview, you can go and say, I am aware of this. Okay. So people want to know whether when you go for an interview, what do they want? For example, I'll tell you a very simple one. When my students from Delhi went to interview to CCMB, Hyderabad, for PhD after BTech. Normally, they don't take students after BTech for PhD. Those days, mine was the first case where they said, you are coming, you must have been trained by Professor Kannan, so we know what his quality is. So they asked them. So first question they said, what is the pH of 10 to the power minus 8 molar HCl? And our kids were pit pat. Whatever question they had the answer, they calculated and showed. And now there are only 5 Ph.D. seats, and all the 5 seats went to my student from one university because they were well trained. So if you are well trained, similarly in Pune University, the first batch, I'm talking of 87, 18 students appeared from my class for CSR net, and all the 18 got scholarship. That never happened in the history because we went through a course program whereby they were trained. And our only idea is how to help you and enjoy learning. I mean, I don't want it to be a torture, so that you are able to benefit. So whatever help I can give, because I have to call experts, I have to pay them. That's the main reason. I can even do it free for you. But for them, I have to pay because, sir, I don't do free. Because there is also a reason they say, sir, if students pay, they are serious to attend. If you do free, they don't bother. That's an impression created for whatever it is. Anything else you want to ask?
Sir, there are
I just want to say one point. Many times what happens in your college, you may not have people who have worked on that area. They are teaching the subject. So I can help you to interact with people who have worked on the area to get more familiar with what you have learned. This is also possible. Yeah, sorry, please.
Sir, there is one question in the message box by
Yeah.
Aryan Yadav. He is asking, is there any scope in astrobiology in India?
See, when I joined biotechnology teaching, there was no scope. We created the scope. So nobody is going to create and ask you, please come. You have to create that. So when we entered the field, everybody said, my father used to ask me, what is this you are talking? I've heard of chemical engineering, I've heard of civil engineering, I've heard of physics, but what is this nonsense you are talking? You understand? It took my father many years to know that I did the wisest thing in my life. Earlier he said, my son, I don't know, is a mad chap doing all monkey things, But the point is, this happens to everybody. And many parents are only familiar with what is around. So you have to create your opportunity. So if somebody is saying astrobiology, you should know whether I can make a career out of it or just academic exercise. Because nobody is going to say there is an opportunity. Opportunity we create. For example, this mRNA vaccine. You must read about Professor Langer. When he started, everybody laughed. It is just not possible. But he pursued. I'll send you the story to you. And today, you have the mRNA vaccine, which was because mRNAs were very unstable compounds. It's so difficult to handle them. I mean, when you purify an mRNA, sometime I will show you how it was purified. They are very unstable. The kind of glassware you use, because there is always a RNase contamination. So the RNA will break down. So how do we do? Like when we isolate a protein, we have to be worried about protease or not there. So how do we inhibit the protease before we purify? So these are all things when we discuss, I'll tell you. So handling biological samples is a very, very big challenge. And that is why I said biotechnology is making a product stable for 3 years or more. Right now, what is happening to mRNA vaccine? It has to be stored at very low temperature. So that is the biggest problem. If I am able to stabilize it at room temperature, I am in business because I can transport it. In India, we don't have cold chains. So many things have to be created, and that's what the government is trying to. For example, if you look at Amul, they collect milk. They made chilling chambers all over the villages. So then the milk is kept cold, then the van will come, it will put it and rush to the processing plants. But these are all stories. You must know that what infrastructure we have, what we don't have. That's why people say India may possible nahi. You understand? But it isn't true because somebody would have invested time. Now, a lot of entrepreneurs have come with material whereby it can be kept cold for 48 hours during transport. You are seeing all these delivery boys coming in the cases, no? There is some material inside which maintains the temperature, keeps it hot or keeps it cold. Yes or no? They're all technologies developed. Yeah.
Sir, there is one more question. The other question is, which is important, field work experience or a PhD degree?
See, nowadays what many people are doing, they go as a research assistant to a lab. They are paid. They first taste whether it's worth doing PhD. Okay? Because a lot of them take a very cool decision, and once they make I think I can do it. Because PhD is an uncertain item. Because if you want to do quality work, you have to discover something. Okay? So that is a challenge. Now, do you want to go through it? Because the companies want people who will do the routine job. Industry, what is it? They don't want creativity. If you are in quality assurance, morning to evening, you keep on doing the same job. Okay. If you look at industry, what is it? There is a research one. There is a development unit. Then there is a quality control. Then you have a toxicological unit. They check whether the compound is toxic or not. Then if necessary, they will go through a clinical trial, preclinical trial, then human trial. Data is collected, analyzed, data analysis happen, and then submitted to the regulatory agency. They go through it. Then they give you permission to go from phase 1 to phase 2. This takes about 10 to 12 years. Now we are trying to reduce it. Like in COVID vaccine, it got cleared in a shortest time because parallelly they did everything. Okay, we have never seen vaccine being approved in 6 months and 8 months ever before. These are all new things because parallelly you do all the tests. Phase 1, phase 2, phase 3, everything runs parallelly. So, in fact, there is a video in my website which you can see if you are interested. There is one on Louis Pasteur and the current vaccine program. One of the experts, one of my former students, gives a 1-hour talk on that. How nowadays vaccine is getting approved. So you should be aware because today technology has advanced a lot. So you will soon see that genetic engineering is nothing great to know. In my time, it was great in 80s. But in your time, if you don't know, it is like saying, I don't know, computer is laughed. You are supposed to know. So saying, I want to be a genetic engineer, nobody says now. At least if you go to an India, then they think you are too old-fashioned as a student. So things have changed. The biggest areas are things like imaging technology or predicting protein folding, so that I can create a new enzyme. These are all the things people are talking about. So it is not, the bioinformatics which is taught is very base level. The real bioinformatics is predicting a structure without doing crystallization, without doing X-ray. Now there's a whole thing called cryo-electron microscopy, which is replacing X-ray crystallography. Okay. So when we discuss, I can take you through each one, how that technology has improved over the years. And today you are in that kind of a phase where things are advanced too much. So what you are learning in college is okay to pass an exam or get a degree, but you should know something else. And that's what I'm trying to help people to be aware of it.
Sir, another question is, please share about epidemiology and its scope in our country.
Well, we have a National Center for Disease Control who run a master in public health. Okay, epidemiology and all. You can look at the NCDC site. Okay, I used to be their member before, so I can send you that. Go and look at that site for this thing. Okay. So epidemiology is certainly a very important area, no doubt about it. But you must know a lot of statistics and all that.
Another question is, after completing BSc, what can we do? I don't know if it is job related or studies, but there is a question. What to do after BSc?
Okay. One is, after BSc, if I were you, if I could get some quick skill and get into an industry, nothing like that. Because the advantage you have is that the industry likes people with less qualification. If you are a Ph.D., they are not very happy, because the owner of the company is quite not that educated, so he doesn't feel comfortable. So you will see there's a lot of psychological barriers. I have experience, I can tell you that, because I worked in industry, so I can tell you that. So they feel more comfortable with people with lesser degree, but skill. If you are skilled, they'll buy you. I have seen people with BSc degree heading the division, because their practical experience is so much. So it has its own advantage, disadvantage. Initially, you may start on a low salary, but as your skill starts improving, people will come and pick you up, paying you higher salary. With PhD, you are overqualified. So what happens? Your job opportunities are restricted. As a BSc, you can diversify much more. Yes, Nishita, you want to ask
Sir,
something?
I'm Tejaswini speaking. Sir,
Tejasvi,
I have one doubt that
ah.
after completing BSc, what we can do? Can we go further and doing MSc or PhD? And how much salary does a biotech student get after joining any company?
See, let me tell you, when you are a raw hand, they don't pay you much. Salaries are very low. But once they
Okay,
find
sir.
you are good at your work, what they want, they will raise your salary. They won't like to lose you. For example, say
Okay.
quality assurance. Let me tell you quality assurance. If you do a good quality assurance and the product doesn't fail in the market, earlier they'll say the quality assurance fellow released a product which is not good and it failed in the market. It's a huge loss for them. They have to recall all the products. So if you have been clearing products very strictly in the quality assurance and no product recall has happened, you are a very valuable QA expert. They will hold you with in a silver plate, because you are very important. So many of them who do well are people who are very good at their hands. You understand? So ultimately, it is not the degree or qualification, but your quality of your work and how much trustworthy is your quality of work. That if product doesn't get recalled, the company is very happy.
Sir, in which companies do we get the job? Like after doing biotechnology, BSc and MSc in biotechnology, in which companies do we get
Look,
the job?
not any. It can be a food industry, it can be pharmaceutical industry. If you look at that list of the government side, there are so many companies in each area. As a BSc, you are open to get into any one of them. So look at each area, they'll show you what are the industries available. Okay. So you don't have to look at
Yes, sir.
which everybody is talking. You can be in agricultural one. It could be veterinary product. It could be any or not. You understand?
So,
Okay,
So
sir.
because you are going to compete with others. Mind you, there will be a BSc zoology student also, botany, chemistry, everybody will be there. Now, how you perform in the interview decide. Like, for example, say BRC has a training school. Are you aware of it? Bar training school. You can
Yes.
appear after BSc. If you do well in the interview, you start off on a very high salary. I know a lot of people who got through the training school. So it is up to you. But from first year, you have to plan. I want to get in here. What should I plan? What should be my? I told you, no. Jayashankar, who is minister now, those days, I know the whole group which used to prepare for the civil service. Every day they'll meet for half an hour, discuss, learn to, prepare for the exam, what is important point. All used to make notes, and many of them appeared and cleared it in the very first attempt because they prepared for 5 years. So very first shot they cleared. So you have to start planning now. Not after BSc, what shall I do? Am I right? Are you getting what I'm saying? So let us plan right from now. Yeah. What a question? Yeah.
Sir, like you said previously, what type of skills do we need to pursue?
The skill will see. The skill comes. We'll have a separate session on various skills for various industries, because each industry has its own requirements. Suppose you are doing downstream processing of say some protein or something. So how to do separate? What are the technologies available? How to optimize that? What is the principle behind it? When to use an HPLC? When to use what equipment? Understanding is required. Now, until you are expand, what is the principle behind it? Once you appreciate, you yourself will say, why not I do this? So until you are exposed to the technique for characterization, because the product purity matters the most, especially in biological purposes, it should be 99.9% pure. You understand? One impurity can create, the most famous, have you heard of Thalidomide story? Have children heard of Thalidomide? Please go and read about Thalidomide. See what happened, a company in Europe, the women during pregnancy have morning sickness. So this company came out with a product called Thalidomide. Okay. And everybody felt when they took it, they felt very comfortable. It was not approved in USA. In Europe, it was approved. But when the babies were born, for many people, the babies were deformed. Some of them did not have nose, some of them didn't have a ear and things like that. All developmental abnormalities happen. And because of that, the company had to rehabilitate it. They made a lot of money, but all the money went in rehabilitation. Read about thalidomide. It starts with a P, PTHA. Please read about it. It's a 1950s story. After that, only clinical trials became very important. Before that, people used to send their drugs immediately. No much trial. Some results are positive, the drug is in the market. Read about sulfur drugs. They're all, before penicillin came, sulfur drugs used to be. People used to make it in their house and supply. And one company ran into problem because some people died. So safety is a very important one. So safety now, so there are a lot of safety regulation. And so a lot of new topics which have come. One is regulatory, another is patenting. A lot of things have come now. You understand? So, but then, I can't finish everything in this 1 hour talks. So we need to have a separate, see, it's a continuous process. It's a continuous learning. This 1 hour business doesn't help us. That's why I said, if you form a club and we meet regularly, one day we pick up, then you can ask all your doubts. Then you can go and read up. For example, have you done microbiology in one of you? Have you? Petri plate, have you?
Ma'am, sir, they had.
They
Sir,
have.
they had.
Okay.
They have microbiology. Yeah.
Okay. How did this idea come? Do you know? Petri plate. Petri is the name of the guy who made that plate called Petri. That's why it's called Petri. Okay. He was a Robert Koch student. Okay. Do you know the story of it? How did this idea come? Okay. Robert Koch was working on, he's the guy who discovered TB, CB, and all that organisms. Okay. So one day in his lab, he had left some half-cut boiled potatoes in the lab. So on the weekend and next day morning, he came on Monday, he saw some spots on the potato, single cell. So he took that one spot and looked under a microscope, and he found they were all homogeneous. So he discovered that to grow microorganisms, because Louis Pasteur said, organisms cause disease, but he had no proof which organism. He did not have a method. Whereas Koch, what he did, he discovered that this wet potato, boiled potato, was a good medium for bacteria to grow. So whichever spores came and settled due to the dust, they were growing as colonies. So he took the colony and examined it. He said, wow, this is. Then he said, I must. So he tried various gels, all these things he tried. And then this thing, Then Petri came to his lab and worked further. Then he developed autoclave the whole thing, put the gel, streaked the thing, then closed it, and then you do. So Petri's idea came out of Robert Koch's observation. So Koch was the man who showed bacteria cause disease, virus cause disease. These are all Koch's this thing. He was the man who said this is the organism. Louis Pasteur did not. He claimed that microorganism cause disease, but the proof came from Robert Koch. But these are not taught in your microbiology. Because they're very interesting. Many of them are chance observations.
So it's there, it's in SY, infectious disease.
Okay,
It's in
they will
SY.
learn. Okay. Now tell me why Edward Jenner is so famous. You heard of Jenner, no? Any one of you? In the vaccine world, he was the first guy to develop vaccination. Okay. Mind you, that method we did, even Indians knew even before he did. But what was so great about Jenner, which others didn't observe? The interesting is that, you know, the cowpox virus he injected into people. Then after a month, he took the live virus from a person with the smallpox and injected, and the person survived. So that second injection, the booster dose, gave a better protection. That was his contribution, that you have to do 2-dose injection. Now we are talking of in COVID third also. Probably we'll talk of fourth also, I don't know. But what I'm saying, Jenner's contribution is doing he didn't know what was happening. He didn't know any immunology, nothing. It was just a chance observation. After 1 month, even Louis Pasteur used to do those things. So what I'm saying is, they knew no immunology. They didn't know what is a T cell, B cell, nothing they knew. But sheer observation. Okay, so see, the idea of forming a club is to keep on talking so that you become familiar. Not be, oh, what a great idea it is. Nothing great, it's chance observation. If Alexander Fleming discovered penicillin, not that he wanted to, it was he observed, but he could never commercialize it. Only Florey and E.B. Chain went about commercializing. Even though, in fact, many people question whether Alexander Fleming deserved the Nobel Prize. That's a point of debate. Okay. So these are all stories I can share with you. Because once you get, then you should know what book to read. You should have some choice of books to read. So this will only come through interactions. So if those of you interested, I'll be more than happy. And I'll get you experts to come and talk to you. Might be from USA, might be from UK, from Switzerland, wherever. That's not the issue. But if you have interest, we provide you people to interact with. Okay? Any other questions? Yes, Nishita? Yeah?
Yes, sir. Sir, actually, I wanted to ask one thing. Sir, if you want any more information, can I email you?
Yes, definitely.
Okay, sir. Thank you.
That's why I said, let us form a club kind of a thing so that we keep, and you can keep on raising questions, because every question has an answer and every answer has a new question. Okay. So what I suggest, see, it is a continuous process. Let me tell you a very interesting incident before closing. When I started this biotechnology program in Delhi, 1999, children got joined, and one boy joined the program, and after 6 weeks, he got selected into Madras Medical College for MBBS. But by that time, he was so fascinated listening to me. He said, sir, I don't feel like joining MBBS. So I said, it's a choice is yours. So the fellow went and said, I don't want the seat. So they thought he was a mad boy. He worked very hard to clear that exam. Okay, and he got in a good CBSE seat he got. He didn't join. So he said, no, sir, I've decided not to join. I want to continue learning biotech. Now you won't. So his parents got very angry with me. What did you do to my son to this thing? I said, meet me after 4 years. We will discuss. Then what I did, since this boy was interested, I said, you must work on brain. So Nature had started bringing out a new issue of Nature Neuroscience. I'm talking of 1999. And I was the head of the library. I was the chairman of the library committee. So I bought all the Nature and I made from first year students go to library and read. And this boy will spend time every day evening, 5 to 8, going through Nature. In the 4th year of his program, there's a very famous university in Germany called Göttingen University, which has produced 43 Nobel laureates. So they wanted to select children for MSc Ph.D. program in neuroscience. This boy went and wrote. He was the only boy who cleared from India. So they invited him to Göttingen for interview. And he stood worldwide first. Then he did his PhD there. Then he did his work in Johns Hopkins University as postdoc. And now he's a professor in Heidelberg Medical School. Hardly that. And doing brain. He said, sir, as an MS PhD, I opened the brain and looked at the brain. I have measured the action potential. I've done a whole lot of things. Sir, I never regretted I didn't do MBBS. I learned and today he is one of the leaders in a particular branch in neuroscience. He talks about calcium signaling. So what I'm saying is, it is your passion. Now, if you listen to others, you would never have done this. And you would have been rotting as a doctor, would have taken him 13 years to become a doctor in the field, because you do MBBS, then you do MD, then you do super specialization, then you become somebody. It's a 13-year program for an average person. But in 13 years, he has become a professor. So it is your faith, it is a belief, your conviction. So there is no set path. The way people say, will I get a job? That is a very middle class thinking. Think big, yeah. I think you will reach the sky. Okay. But you should know what all should know, which books to read. That boy used to read regularly Nature. So when he went for interview, they said, who is the professor who has taught you? Indian children don't talk like you do. Who taught you? They asked. So what I'm saying is, they also find out that who could have taught like that. So it makes difference. So you are in the right stage. You have another 2 years. Plan your career now. Look at the website, which are the areas, which are the institute. We'll work out a summer training for you to go there, learn more, interact with people, or go there, talk to the scientists, what do they do. There'll be a lot of people. You see, in India, people love to talk. Only you should have the you should have the patience to listen to them. So I always say you should have big ears and small mouth. You utter one question, they'll give you 10 answers. Enjoy that. Okay. So don't think, see, pursue what you like, the rest will happen. So that is what my experience. Okay, shall we close it?
Yeah, I think there are a few more
Yeah,
questions.
please ask.
Shruti, what we can do is
No,
we can collectively put it in.
one or two, just I'll reply now. Tell me.
Okay. Someone wanted to know about pharmaceutical biotechnology.
See, these are all words. Agriculture, biotech, pharmacy. Only thing, see, pharmacy, if you look at, today the hottest selling products in biotechnology are therapeutic antibodies. If you look at the top 15 selling products in biotech, 10 of them are this thing. Now, this is made by a technology called phage display technology. Okay. And this was discovered by one of my seniors, one of my old friend, called Greg Winter, who got Nobel Prize in 2018. Okay. And he created all it's a very beautiful technique to do. Okay. And by this, he developed first this thing for rheumatoid arthritis. Okay. Well, the drug is called Humira. Please go and read about Humira. Humira. Okay. So when we say pharmaceutical biotechnology, there is no more antibiotics or bulk drugs. As far as the biotechnology is concerned, these are all proteins. Okay, and read about that. Read about Humira. Then you will realize what a huge field. In fact, I'll tell you another very interesting thing which is coming is what is called PROTAC. That is PROTAC. Okay, that is, you know, people are now knocking out genes to see the function. Okay, by this technology, I can knock out the protein without disturbing the DNA or RNA or anything and see what happens to it. Because the problem with gene knockout, if it is lethal, the cell will die. But in this, only you are removing that protein. You degrade that protein and see if it is not there, what happens, or if it is there, what happens. So it's a new approach. This is called the PROTAC technology, which just came in 2020. Interestingly, I used to talk of this way back in 1986 with my MSc students. So now they recently said, do you know, you taught us all that. I wish you had continued. You would have yourself discovered this. But what I'm saying is, it is very important to that technologies are shifting. It is no more DNA. Now, PROTAC is a hotly debated topic. Okay. Because if one protein level goes up, which is giving problem, just remove it. Don't disturb the rest. You understand? DNA means you will create a mutation. You will have a lot of other things. But here, you just remove the final product. So this is a new approach, which is so every time new ideas are coming. Is that clear? So a lot of things like that. I mean, I can endlessly go on. Any other question?
The other two, I think you have already answered. The one is about job opportunities, and other is about joining the club. I think
See,
the
yeah.
it itself gives the amount of
Job is something you create for yourself. Don't I wait, because they are looking for people with a pair of hands. Don't become a pair. This is what is happening in our IT field. We are called the IT fellows are called the cyber coolies. You know, like in the railway station, people carry luggage. No, so in the IT world, we call them cyber coolies. So coolie anybody can become. If Indian can become, so can a Chinese or somebody else. So that's a huge crowd. That's why I used to tell long time back, many of us work from $0 to $15 per hour work. Americans do $300 to $400 per hour work. So you can see they enjoy, they go, they enjoy, party, and make you all work. Our boys are spending sleepless night working in the various call centers and all that. So I think I don't want biotechnologists to become like that.
yes. I think that's it. Shruti, can you please ask one student to propose the vote
Yes,
of thanks?
yes, ma'am. Nishita is going to propose the vote of thanks.
Yeah.
Towards to
Nishita.
you, Nishita.
Yes, ma'am. A very good evening to everyone. It gives me an immense pleasure to deliver the vote of thanks for this event to dignitaries assembled here. I would like to thank our chief guest who honored this session with these inspirational thoughts. We are glad to have such inspiration as you in our life to guide us through the path we want to choose. My heart fills with lots of gratitude and respect for our distinguished guest, Mr. Kannan Krishnamurthy sir, for not only sparing his valuable time for us to grace this event, but also for enlightening us with the commendable talk on the subject. Thanks a lot for clearing our concepts and enhancing our understanding pertaining to the biotechnology. You have indeed put best of your efforts to make this event an unforgettable event. We really owe you a lot. Also, I would like to thank our beloved professors, volunteers, and last but not the least, our beloved audience for making this session as a grand success. Once again, I thanks one and all present here. Thank you so much.
Yeah, I would like to dedicate this session to Dr. Sneha, you know, your former head of the department, whom I was fortunate to talk to her. Actually, we both have been planning to organize this at that time. Unfortunately, the destiny didn't allow us to meet earlier, but thanks to Indu, you know, we could meet again. So I look forward to any time. Feel free to write. Thank you so much.
Thank you so much, sir. Thank you so much for your valuable
Let,
time.
let everybody switch on and let her take a photo, I thought.
Okay. Students, please switch on your camera. Shrushi, please do the needful.
Yes, ma'am, I'll do it.
Yeah.
At least smile, please.
Yes, ma'am, done.
Thank
Done?
you.
Yes,
Thank you
thank
so
you so
much.
much.
Thank you so much, sir.
Okay.